In order to prepare for expression of array-immobilized cDNA, printed slides are first placed in a shallow incubation dish with the printed face up and blocked with 5 mL of blocking buffer, ensuring sufficient buffer to completely cover the slide, and then incubating with shaking for 1 hour. While the slides are blocking, we thaw the components of the cell-free expression system, the rabbit reticulocyte lysate, on ice, and assemble the other materials that we require for expression of the slides. A pair of forceps, a small wooden spatula, and the slide gaskets that seal the print face of the slide, allowing coverage with the rabbit reticulocyte lysate. Once the slide blocking is complete, the slides are removed from the incubation dish. Excess blocking buffer is rinsed from the slide using a bottle of Miersch water, and then blown dry, again using filtered compressed air. The slides are then sealed with a plastic slide gasket that enables the surface to be covered with the cell-free expression mixture. The slide gasket is carefully placed to ensure that the printed surface of the slide is not disturbed, facilitated by placing the slide on a raised surface and then aligning during placement. We then aim to express the slides using a rabbit reticulocyte lysate cell-free expression system. This coupled in vitro transcription translation system requires that several components be mixed together prior to slide application. DEPC treated water, 2 amino acid mixtures, each lacking a different single amino acid, a reaction buffer, the T7 polymerase, and the rabbit reticulocyte lysate that contains the ribosomal components required for mRNA-directed protein assembly. Components are combined according to the manufacturer's instructions and carefully and adequately mixed to avoid bubbles, but to ensure a uniform and homogeneous mixture. The mixture is then applied to a gasket-covered slide, first ensuring that the gasket is properly sealed by gently running a thumb down all edges of the slide. 135 microliters of reticulocyte lysate is then quickly added to the slide via one of the gasket inlets, gently massaging the surface of the slide to ensure an even distribution of the lysate over the entire surface. Any areas with bubbles or not covered during lysate injection can be removed by gently tapping and massaging the surface of the gasket, directing air toward the gasket inlets, ensuring even coverage of the slide. We then apply gasket seals to both inlets to stop evaporation of the lysate during incubation and expression, and then slides can be placed in a temperature-controlled incubator that has been pre-warmed to 30 degrees Celsius and incubated for 90 minutes at 30 degrees Celsius, followed by a 30-minute incubation at 15 degrees Celsius. Following incubation, gaskets are carefully removed from the slides and the slide rinsed to remove residual reticulocyte lysate. First with 5% skim milk in PBS with 0.2% tween, and then again 3 times for 5 minutes with a 5% PBS-T solution, followed by a 1-hour blocking step during which time the slides are gently rocked at room temperature. Following completion of the blocking step, excess buffer is removed from the slide, after which they can be stained with anti-affinity tag antibodies to determine protein expression, capture, and display, or they can be stained with human serum to detect antibody interactions with proteins on the array. We will demonstrate the staining and development of slides with anti-affinity tag antibodies. Primary antibodies are prepared at an appropriate dilution in 5% milk and PBS-T, and 2 mL of that solution carefully added to an expressed and blocked slide in a sealable slide chamber. The slide chamber is then sealed and immediately placed in a secure fashion on a rotating slide rotisserie for an appropriate incubation time, allowing sufficient time, whether at room temperature or at 4 degrees Celsius, for equilibrium binding conditions to be achieved. After completion of incubation with primary antibodies, slides are washed 3 times for 5 minutes with 5% skim milk and PBS-T, and then exposed to an appropriate dilution of secondary antibodies, in general for 1 hour. Slides are then removed from the incubation chambers and washed with PBS-T, again 3 times for 5 minutes, in preparation for the final stage development, in which we will demonstrate the use of a tyramide amplification system that fluorogenically immunostains features in an HRP-dependent reaction. We first prepare 600 microliters per slide of a 1:100 dilution of the TSA Psi3 development reagent in the manufacturer's supplied diluent. A washed, expressed, and antibody-stained slide is rinsed with millicue water to remove excess PBST. It is often helpful to wipe the back of the slide and then drain excess water from the front of the slide. We then add 500 microliters of TSA reagent to the slide surface and cover with a lifter slip carefully placed to ensure even distribution of the TSA reagent across the entire slide surface. The reaction is timed and allowed to proceed for exactly 10 minutes, covered, protected from light, after which the lifter slip is removed and the slide rinsed thoroughly with millicue water. We then can dry the slide using filtered compressed air, after which it is ready for imaging. Developed slides are then loaded into a slide cassette, and the slide cassette loaded into the scanner. Slides are then scanned at an appropriate laser intensity, gain, and resolution, and then signal intensity data extracted using image analysis software. In closing, we have demonstrated the purification of plasmid DNA in a high-throughput fashion for the construction of nucleic acid programmable protein arrays. We have further demonstrated the use of cell-free expression systems to convert cDNA arrays to protein arrays for subsequent multiplex experiments aimed at probing functional protein interactions and activities.